So I want to introduce a very special principal investigator who really, I think, models the pathway to translation. That's Jay Chiarini. All of you do, but I want to highlight Jay's work. Picking up from where Bruce Baum left off and all of Larry's and his team's work on basic salivary biology, Jay has forged pathways in understanding how to treat disorders using gene therapy, per se, in collaboration with Blake Warner, who's back there in the last row, performing some very, very innovative experiments. So Jay, all yours.
Thank you, Rena. The work we've done in gene therapy really started under the leadership of Larry and Hal. And the concept of gene therapy has been around ever since we discovered that DNA was a hereditary material, the idea of changing genes to correct a disease. NIH, the intramural program, has really been at the lead of that, at least since the early 80s. And within NIDCR, we've taken the lead for oral gene transfer and correcting diseases with unmet clinical needs in the oral cavity. Specifically, what we focused on is radiation-induced xerostomia. The idea that patients are severely affected by the loss of salivary gland function. Imagine that. It's an important organ, really. And that by understanding the physiology that's associated with the loss of function, that we could develop a therapy to correct that. And as Dr. D'Souza indicated, that work really started by two NIDCR researchers being stuck on an airplane, waiting to taxi, and talking about the important findings that they had at a recent meeting. And the idea that if we could re-engineer the remaining tissue within the gland to have a gain of function, to move water again, then we could develop a therapy for this. And this was about 30 years ago. And that's one of the beauties of the intramural program is long-term stable funding. At least I hope it still is. So that started the question of how can we develop a therapy, and specifically focused on gene therapy, how can we locally introduce the genes into salivary glands? What gene would we introduce? And more importantly, what's a relevant animal model to test this in? So for the past almost 20 years, it took that long to figure out and answer all these questions. My group is focused on developing the molecular truck to deliver genes into cells. And that's been a major component for effective gene transfer, and launched a proof of concept clinical trial in 2009, which was under the leadership of Bruce Baum, which was largely successful and showed that it was possible to re-engineer the remaining epithelia in the gland to have a recovery in function. And in 2016, we launched a safety phase 1 study that again validated, or is validating, the outcome, and has been successful enough to launch a phase 2 study, which is now taken over by a corporate partner and is moving ahead. NIDCR is still involved in that, but it's a multi-center study. So the future of gene therapy in the oral cavity is very bright. What we plan to continue doing is to try and apply this technology to other unmet clinical need, or diseases with other unmet clinical needs, such as Sjogren's syndrome. But the salivary glands are uniquely positioned to produce proteins that go both into saliva through the exocrine pathway and, little appreciated, the endocrine function. Well, here's a natural secretory epithelia that produces 1 gram of protein a day, and it's easily accessible. We can re-engineer them to produce hormones that might be short-lived and not amenable to enzyme replacement therapy, and have them traffic back into blood and use the salivary glands as a depot organ for recombinant protein therapy.
Excellent. Great. So obviously when we target downstream effector molecules, and aquaporin obviously contributes to the water component, and you look at the normal physiology and development of salivary glands, how important do you see is the nerve supply and the vasculature and in restoring function in radiation-induced xerostomia, for example, or even in Sjogren's.
You're absolutely right. There comes a point for our aquaporin therapy to work, we need to have some functional epithelia. That's not always the case. So regenerative medicine, regenerative cell therapy is definitely the next level and the future for glands that we can't restore.
Can I break the rules and ask for a question from the audience? Larry, where would you envision this future, since you were one of the grandfathers of the time? Oh, no, your grandfather. You're the grandfather. Yeah.
I guess next is senescence. Yeah, well, you know, I think Jay captured it well, at least in you know, exocrine endocrine axis. If you look at a number of rare diseases, you know, we're trying to figure out how to do gene replacement therapy. Maybe we should leapfrog it and provide the actual product of interest through an ancillary production system, be it salivary gland, be it muscle, you know, pick whatever truck, you know, as you call it. But I do think that as we get more and more information about some of the multi-gene disorders, that's going to be the real trick. Because right now, monogenic efforts, I think we have at least a roadmap to this, you know, cure, but it's the multi-gene, and I think Marion said it very well. It's context dependent. And so in addition to producing whatever it is that you want, you got to make sure it goes to the right place at the right time, spatially, temporally, and so forth. And so when you see these hairball diagrams that Pam was alluding to, the so-called multi-omics, as daunting as those things are, that's the roadmap to figuring out that four-dimensional problem that, you know, that we're all sort of wrestling with. But I think with the computational power that we have, we're going to get there. And so then you take not one gene, but multiple genes, figuring out, you know, much like you would if you're running a rail station, you know, which comes first, which comes second, where does it get expressed, for how long, and where. And that's when we really begin to focus on some of the most complex conditions that we know. Now, I'm going to let the immunologists tell us about immunology, because no one understands that except immunologists. But you can envision then the interplay, because all we're talking about is host, and that's part of the host, but then you have those messy bacteria and viruses and so forth. So, but that's how that multi-omics approach, I think, gets to where you want to go.
In fact, there is a strong in Sjogren's, of course. Yeah. But I want to thank you, Larry. Thank you, Jay. Thank you very much. That was very